Hello all of you. So in today's video we'll be discussing the solutions for GATE, Excel, and the topic which we are covering today is biochemistry, enzyme kinetics, regulation and inhibition. So in this video we'll be discussing few questions related to the basics of enzymology, about the naming of enzymes. So for the course, you can refer to the BT05 Enzyme Science and Technology course of NPTEL. Okay, so to start, if we'll talk what are the enzymes. So an enzyme is a biocatalyst. So an enzyme is a biocatalyst which changes the rate of the reaction, okay, without being utilized itself in the entire process. Okay, so if any substrate which is getting converted to the product, so with the help of enzyme this reaction gets speeded up many times. Okay, and the conversion of substrate into product will be much more faster. Okay, so how does enzyme do that? So enzyme do it by reducing the activation energy. So if this would be a simple graph from, this is the, okay, so this is the point of the substrate, this is the energy level of the product, or if it needs to be reached from substrate to product, so how the reaction takes place. So substrate, uh, enzyme plus substrate, it will form an enzyme substrate complex, and then enzyme will get released and substrate will get converted to the product. So in between this enzyme substrate complex is formed. Okay, so generally this reaction will take place like this. Okay, substrate needs to get converted to a transition state, and then this transition state, which is highly unstable because of the higher energy, it gets converted to the product. But this is without the enzyme. So generally without the enzyme it takes lots of energy to reach to this transition state. Okay, so the energy barrier is very high, which is there for substrate to cross. But what does enzyme do? Enzyme reduces this transition state energy barrier. Okay, so it brings it here, okay, somewhere, and now it is easier for the substrate to cross this energy barrier and gets converted to the product. Okay, so this is the like reduction in the activation energy which is caused by the enzyme. Okay, so without enzyme this was the activation energy which needed which substrate was needed to get the reaction initiated. But without enzyme, with enzyme, now only this much activation energy is required for substrate, which is a much more easier process than reaching from here to there compared to from here to there. Okay, so this is the activation energy with enzyme, without enzyme. Okay, so that is what an enzyme basically do in a reaction, chemical reaction. Okay, so next if we'll talk the enzyme structure and general classes. Okay, so to generalize So enzyme, we all know enzyme, basically all the enzymes are proteinaceous in nature. Okay, except we have exception. We have exception where catalytic RNA molecules are also there, which are called as ribozymes, which also work as a catalyst or biocatalyst. Okay, so except ribozymes, all other enzymes are proteinaceous in nature. Okay, so we'll start dividing. So enzyme could be of two types. One is if I'll say simple enzyme, okay, only made of protein. Another comes conjugated enzyme, okay. So in conjugated enzyme, uh, that means the enzyme is joined to a non-protein part as well, which non-protein part is called as cofactor. Okay, so in conjugated enzyme, generally the enzyme is in active form only along with its cofactor. If in a conjugated enzyme, if we remove the cofactor, the remaining enzyme will be called as apoenzyme. Okay, so conjugated enzyme minus cofactor will be called as apoenzyme, and with the cofactor, and it is apoenzyme is the inactive form of the enzyme. Okay, it will be active only when joined with the cofactor, and after joining with the cofactor, the entire active enzyme is called as holoenzyme. Okay, so in cofactor, generally we can have two types of cofactors. One is like simple metal, or complex organic molecules, for example vitamins and other things. Okay, so these are called as, so this complex organic molecules are called as coenzyme. Okay, so in simple metal also it could be two types. One, we can say metal activated enzyme, other one is metalloenzyme. Okay, so here what happens, and the enzyme binds to metal. In this case, the enzyme binds to metal loosely for the activity, while metalloenzymes are tightly bound to the metals even in its native form to um, stable the structure, and that's how the enzyme is functioning. Okay, so here the loose bonding with the metal, here tightly bound metal is used, and enzyme remains like that only in the native form, the metalloenzyme form. Okay, then comes to the complex organic molecules, which also we can refer as coenzymes. So there also we have two types. Okay, one which is so these coenzymes are joined to the enzyme with loose or tight covalent or non-covalent bonding. So if it is tightly joined with the enzyme via covalent or non-covalent bonding, it is called as prosthetic group. And if it is loosely bound with the enzyme, then it is called as co-substrate. Okay, so these are the different terminology which is being used when it comes to the enzyme. Okay, so now it is clear from this slide at least, okay, how you can divide enzyme into subcategories. What does a cofactor means? What does a coenzyme means? What is a apoenzyme, haloenzyme, metalloenzyme, metal activated enzyme? Okay, so all those things can be clear from this slide, I guess. Okay, so moving ahead. So okay, so usually many enzymes have some common names like trypsin or some uh, some type of reaction on the type of reaction they catalyzes, and just putting a suffix to it. Okay, so those are the traditional naming of the enzyme, but later, as the more enzymes are being added to the list of the enzyme, it is difficult to give a generalized name to all. So for the naming of enzyme, the Enzyme Commission has developed a rule for naming of enzymes. Okay, so according to this rule, each enzyme is classified and named according to the type of chemical reaction it catalyzes. Okay, so the Enzyme Commission has given each enzyme a number with, so Enzyme Commission, it's also called a key. Easy. So it has given each enzyme a number. Okay, so according to that, the enzymes are divided into 7 classes. Okay, so first class, like EC1. EC1, we have oxido reductase. We have EC2. EC2 is the transferases. EC3 hydrolases. EC4, where comes all the lyses. EC5, where comes the isomerases. We have EC6, in which we have ligases. We also have a new category which are called translocases. Okay, so these are the 7 categories of enzyme according to the Enzyme Commission, and these are the first Enzyme Commission number. Okay, so the Enzyme Commission number is generally of four um, 4 digits, you can say, in or in 4 parts. Okay, so uh, like for hexokinase, the Enzyme Commission number is 2.7.1.2. So the first part defines the major class. Okay, the major class, which being 2 here, is the transferases. Okay, so this is the major class. Okay, or then second digit defines the class, third digit defines the subclass under it, and the last one is specific for the enzyme, or you can say it is the serial number in the subclass indicating the order in which the enzyme is added to the list. Okay, so that's how you can divide and enzyme commission naming of any enzyme. Okay, so here we'll start talking about the major class and the functions and the type of reaction being catalyzed. So EC1, which is oxido reductase. Okay, so as the name suggests, oxidoreductase catalyze the sorry catalyze the oxidation and reduction reactions. Okay, so for example, if two substrates are there, oxidized and reduced, with the help of this enzyme, one gets reduced, another one gets oxidized. Okay, so these enzymes generally used as a electron donor or acceptor. Okay, so if the reduction is taking place, that means acceptance of electron, acceptance of hydrogen, or removal of oxygen. Okay, if oxidation, that means removal of electron, removal of hydrogen, and addition of oxygen. Okay, so that's what happens in this kind of reactions. Okay, talking about few examples. So if we talk about few examples, the enzymes comes under these categories are oxidase, dehydrogenase, oxygenase, and peroxidases. Okay, so here we can see there are two oxidases. Okay, both involved of involving oxygen in the reactions. So if we'll talk about the difference, okay, so oxidases, what does they do? They use oxygen as an electron acceptor but do not incorporate it into the substrate. Okay, oxygen as electron acceptor. Talking about oxygenases, so so here they this enzyme incorporates oxygen into the substrate. Okay, so there is one enzyme, for example, that is monomethane oxygenase. Okay, so here this monomethane oxygenase, so here this oxygenase incorporates oxygen into the substrate. Okay, okay, it incorporates the oxygen into the substrate, so that is an oxygenase enzyme be involved and carried out the oxidation reaction, oxidation of methane. Another is the oxidase, which act as an electron acceptor. Okay, so here oxygen accepts the electron and allow the reduction reaction or oxidation reduction reaction to take place. Okay, but here in oxygenase case, it incorporates the oxygen into the substrate. Okay, moving ahead, next class is the transferases. So transferases catalyze the reaction that involve the transfer of groups from one molecule to another. Okay, so these group includes amino group, carboxyl group, methyl group, phosphoryl group, and etc. Okay, so the basic scheme here is, so this involves the transfer of groups. Okay. So for example, we can say like trans carboxylase, which involves the transfer of carboxylate group to a substrate. Trans carboxylase. Okay. So transaminase are also the part of this group. Transaminase, which transfer of amino group. Carboxylate group. There are kinases, which transfer of phosphate group. Okay, we also have phosphorylases, which also involves the transfer of phosphate group, but kinase does the transfer of phosphate group from ATP to another substrate, and phosphorylase transfer the inorganic phosphate group to a substrate other than ATP. Okay, they do not use ATP for the transfer of phosphate group. Okay, so these are the some examples of the transferases in the basic scheme. So for example, kinases, we have hexokinase. Okay, and similarly all other amino acids, transfer of the groups via the enzymes. Okay, so next class comes the EC3 enzyme commission, third major class, which is hydrolases. Okay, so in the hydrolases catalyze the reaction in which the cleavage of bonds is accomplished by adding the water. Okay, so if this is the bond which needs to be break, they use water to do the hydrolysis reaction. Okay, and that's how the bond is being broke. Okay, so here we have many different enzymes. For example, phosphodiesterase, which cleave the phosphodiester bond. We have phosphatases, which cleaves the or remove the phosphate from a substrate. We also have peptidases, which also leave amide bonds such as peptide bonds in the proteins. Okay, so all the digestive enzyme like uh, um, alpha amylase, okay, pepsin, all these are the hydrolysis group. Next group comes the EC4, which is lyses. Okay, so lyses are the enzyme that catalyze the breaking of the bond. For example, carbon-carbon bond, carbon-oxygen bond, carbon-nitrogen bond. Okay, and other bonds by means other than hydrolysis and oxidation. Okay, so these bonds are like cleaved by the process of elimination and result in the formation of double bonds. Okay, so we'll see the scheme. Okay, okay, so these enzymes cleaves the bond and result in the formation of double bond. Hmm. Cleaving the formation of double bonds. Okay, like this. Okay, and same the reaction. Okay, and if the reaction, which is the same reaction, if takes place in the reverse, it is the enzyme is called synthase. Okay, okay. So in the forward, when the double bond is being formed, it is called as lysis, and if the same reaction is taking place in the reverse, it is called as synthesis. Okay, so for example, aldolase, aldolase, removal of aldehydes by elimination reaction, synthesis, many syntheses are involved, like link two molecules without involvement of ATP. Okay, um, decarboxylases, the removal of CO2 as an elimination reaction. Okay, so these are the example of lysis. Okay, next comes the EC5, which we can say as isomerases. Okay, so here the isomerase catalyze several type of intramolecular rearrangements and yields an isomeric form. So it is, okay, so it is like intramolecular rearrangements. Okay, so under this category comes all the mutases, cis trans isomerases, and we have epimerases, okay, and racemases. Okay, and these are the examples of isomerases. Okay, and we also have EC6, which are ligases. Okay, ligases are the ligase that catalyze the bond formation, carbon-carbon bond, carbon-oxygen bond, carbon-sulfur bond, all these bond formation, and the energy is required for this bond formation, which is supplied by the ATP hydrolysis. Okay, so bond is being formed by ATP hydrolysis. Okay, this is the scheme. So all the synthesis comes in this category. Okay, carboxylase is also example of this group only. Mm. Other than that, we have the new class of enzyme, which is EC7 translocases. The translocases catalyze the movement of ions or molecules across the membrane or their separation within the membrane. Okay, so these type of enzymes are come under this new class of enzyme, which is also called as translocases. Okay, so this is the summary where we have read about the seven enzyme major classes. We have also translocases here. So oxidoreductase, which carried out the oxidation reduction reaction, transferase, which is involved in group transfer, hydrolases, which cleaves the chemical bond by hydrolysis, lyases, which is a non-hydrolytic cleavage of the chemical bonds, isomerase, isomerization of substance into structural or optical isomers, ligases, joining of two molecules by forming new covalent bond using the energy from A to P. And the other class, translocases, which catalyze the movement of ions or molecules across the membrane or separation or their separation within the membrane. Okay. So if we see the questions, so this question has come to the 2018 GATE Excel question paper. So the question states, which one of the following classes of enzyme does chymotrypsin belongs? So chymotrypsin, we all know it is a peptidase enzyme. Okay, so it is a digestive enzyme. So all the other enzymes which are like oxidoreductase, it is not true. Transferase is also not true. Isomerase is also not true. So chymotrypsin carries out such a reaction which involve the hydrolysis of peptide bond using water. Okay, so chymotrypsin comes under the category of hydrolases because it cleaves the peptide bond. Okay, it cleaves the peptide bond. Okay, the C terminal and the carboxyl terminal of the aromatic amino acid, there it makes a cleavage, and by addition of water, so the peptide bond gets broken. Okay. Next question is, which enzyme catalyzes the oxidation reduction reaction directly incorporates oxygen into the substrate? So we just now read that oxygenase directly incorporates oxygen into the substrate, while oxidase work as an electron acceptor and do not incorporate the oxygen into the substrate. Lyase, a class of enzyme that catalyzes. Okay, so hydrolysis reaction, no, that is done by hydrolases. Group transfer is done by transferases. Oxidation reduction done by oxidoreductase. And then the addition of groups or to double bond or vice versa. Okay, so this are the classification or the definition of the lysis. Okay, addition of groups to double bond or vice versa. Okay, like breaking of bond and then creating the double bond. So that is the function of the lysis. Okay, thank you. So thank you for listening.